Hey, biology students. We're going to take a couple minutes here and go through a summary of DNA replication that you can have 24/7 access to, as opposed to just the time we get to spend covering it in class. I need to start off by saying that this image is not mine. I did not create it, but as I made the PowerPoint that this is on 2 years ago, I don't remember where I got it from and I couldn't find it. So hopefully I'm not violating any copyright laws, but this is not my work in this image. However, the explanation that follows is original and it is mine. So here you can see, and for those of you that are not in my class, I'm assuming that you already know a little bit about DNA replication. So we're going to go walk through this first by following what happens in the leading strand, shown here on top, and then the lagging strand on bottom. So let's start off with noting the template strand orientation in the leading strand. Notice the template strand is traveling in the 3 to 5 direction as we move right to left on your screen. In this image, the overall direction of replication, as indicated down here on the bottom, is actually going right to left. Okay, so when I say the template strand has 3 to 5 orientation, that is this dark blue strand on top. So this is the leading strand now, because as we know from class, the enzyme DNA polymerase can only add sequential nucleotides in the 5' to 3' direction. So since the template strand is going 3 to 5, that allows the synthesized strand to go 5 to 3. In other words, the DNA polymerase can just cruise right along this template strand, adding individual nucleotides as it goes. So here these purple ovals are DNA, it's a DNA helicase complex, which is going to unzip DNA. Okay, so as the DNA gets unzipped, it presents one of the strands, one of the template strands, in order for it to be replicated. Right behind the helicase complex, we have these little red semicircles, which are single-strand binding proteins. They prevent these two template strands of DNA from reannealing. So if left to its own devices, this DNA strand would probably come back together, and that would be really bad. We wouldn't be able to replicate the DNA. So these little red semicircles here, as shown in the picture, are going to hold the two strands apart from one another. So now on the leading strand, somewhere off to the right of the screen where we can't see, an RNA primer would have been added by the enzyme RNA primase, maybe somewhere over here. And as soon as that RNA primer is added, DNA polymerase now has a binding site where it can attach to the DNA and then add nucleotides in the 5' to 3' direction. And recall that DNA polymerase needs that RNA primer because it is a little bit deficient in its, not a little bit, it is deficient in its ability to synthesize DNA if it doesn't have a double-stranded area of DNA for it to originally bind to. And so that's the reason why we have the RNA primase and the RNA primer. And that pretty much covers the leading strand. Once the RNA primer is laid down, DNA polymerase can just follow the helicase complex, and as the DNA gets unzipped, polymerase follows behind, adding sequential nucleotides in the 5' to 3' direction. And now for the lagging strand, it's a little bit more difficult. As you can see in the picture, these lagging strand is actually added in fragments. And so as the DNA gets unzipped, the template strand is actually traveling in the 5' to 3' direction, moving right to left, which means the DNA polymerase cannot add new bases in the same direction. It can't add bases traveling to the left on your screen. It's going to have to travel from left to right. Okay, because when it travels from left to right, it's able to add new nucleotides in the synthesized strand in the 5' to 3' direction. And so as DNA helicase unzips a section of DNA, right behind where it's unzipped, in this area, RNA primase can come in and add an RNA primer, and then DNA polymerase binds right here and can synthesize a small segment of DNA up until it bumps into another one of these fragments. Okay, and so people always ask, well, why is it in these fragments? Why can't it go in one piece? And the reason why DNA polymerase is not going to synthesize this lagging strand in one piece is because if we waited for the helicase to unzip the entire DNA strand, then there'd be a possibility for the DNA strand to fold back on itself, get twisted up. A bunch of things that could be very complicated could happen that prevent us from synthesizing a new strand of DNA. And so DNA instead on the lagging strand gets added in these fragments called Okazaki fragments. And so DNA polymerase will add new bases in the 5' to 3' direction until it bumps into an RNA primer, in which case it will detach and stop. And then later processes will connect those pieces. So hopefully this is helpful, and good luck on your test.